You know very well that living organisms are consisting of cells. Some living organisms, their bodies are consisting of great number of the cells, like human, or animals, or even plants. They are consisting of great number of the cells. This group is called the multicellular. Multicellular means that they are consist of great number of the cells. For instance, the human body consisting of trillions of the cells. And the long trees, the huge forests, the big animals, their bodies are consisting of great number of the cells. These groups are called the multicellular because they are consisting of great number of the cells. On the other hand, some living organisms, their bodies are consisting of just one cell, which are called unicellular. From these examples, viruses, bacteria, algae, fungus, all of them are considered from unicellular, so they can be seen only under the microscope. Your microscope here is used to magnify small things which cannot be seen by the naked eye. And we have different types of the microscopes. Some of them are called light microscope, like this one. This type of the microscopes is called light because it depends on light rays passing through the stage, so that if you look at the specimen here, you can find out what is prepared on the slide. But I have to tell you something, the microscopes, and the light microscopes especially, they have magnification force smaller than the other types of the microscopes. So we have two types of the microscopes, light microscopes and the electron microscope. The electron microscopes have the ability to magnify small things which can't be seen by the eye up till 500,000 times. And the two types of the electron microscope, one of them is called scanning electron microscope, and the other one is called transition electron microscope. So what is the difference between both of them? Scanning is the one which can look at the specimen from its surface only. But the transition, the other type of the electron microscope, which can look through the specimen. We can investigate the inner parts of the specimen easily. So both of the two types of the electron microscope are better than the light microscopes because they give accurate details of the specimen itself. So the microscopes can magnify small things that can't be seen by the eye, like the microorganisms. They are called microorganisms because they can be seen only under the microscope. From the cells that can be seen under the microscopes, some cells inside the human, like human cell or animal cells, we cannot investigate the cells by the naked eye because they are very small, microns. That's why they are called microscopic organisms. And also we can use the microscope to look at the plant cell. Right?
Yes.
Okay. So I want all of you to have a look at the first type of the microscope, which is light one. Light microscope is called the light microscope because we use a source of light under the stage. When we switch on the light and fix a slide which is previously prepared, plant cell or animal cell or even algae or even type of fungus or yeast fungus, as you are going to see and visit the lab right now, you can look using the specimen under the stage, over the stage, and use the eyepiece here to look at the specimen. The arm can be used to hold the microscope, and the base can be used to settle it down so that it shouldn't put on an inclined surface. Why? To be settled in this position, fixed in this position, and the two focus here to change the magnification force. Now, I want all of you to make a simple comparison between the two types of the microscopes. You have a table. Each group you have to share with each other to make a comparison between light microscope and electron microscope. I'm going to give you a couple of minutes. Ready? Steady? Go.
Magnify the specimen 1000 times. Magnetic scope.